Okay, so as I said, I'm Peter Cooper. My email address is there. If you have any questions, you can always write to me, peter.cooper@nih.gov. That's pretty easy. We're going to talk about what I mean by genomic context. That won't take long. And then I'm going to talk about something called the Identical Proteins Group Report and how to see the corresponding genomic regions for BLAST hits. So we're going to use this graphic view on BLAST to do that. And then at the end, we'll wrap it up by showing you some scripting ways to get to some of this information using EDirect and some additional programs. Okay, so what do I mean by genomic context? I mean what genes or protein products or regulatory sequences or other sort of features are either upstream or downstream in the genome for matched proteins. And these are the kinds of things that everybody is interested in because they can tell you about regulatory mechanisms. It's useful to know from a comparative genomics perspective, like, how did this particular set of genes, how did they change over time? Or are there multiple copies in some genomes and not in others? Those kinds of things. And also, another sort of way to use this would be just a quality check on sort of genome assemblies and annotation, because you might say, hey, you know what? This genome is missing this particular gene. Maybe that is a problem with the assembly. So you could look at things like that. So the particular set of genes that we're going to look at today are the ones that are involved in the type 3 restriction endonuclease cassette. This is a defense mechanism that bacteria use, one of them, to get rid of foreign DNA. And this is a typical example of what you see if you look into a bacterial genome. You'll see that there is a helicase at the 5' end of this thing. There is the methyltransferase, which is the thing that methylates bacterial, the genomic DNA of the host, so that it doesn't get cut. And then there is a binding protein. This is the type 3 restriction endonuclease subunit R. And so that's a typical kind of situation. And if you look around, you'll see that there are some changes in this in some kinds of bacteria. But this is the one, sort of the canonical one that we're going to be looking at today. And what we're going to do is focus on the site-specific DNA methyltransferase for the searches today. So we're going to look for that, find it in BLAST, and then take a look in the genome and see if these other two genes are flanking it in the genome that we're looking at. Okay, so if we're doing a nucleotide search, this is actually a fairly straightforward thing to do. Now, what I mean by nucleotide searches, I mean the database you're searching is nucleotide. Because what we're doing is we're looking down in the alignment section. And when we have a tBLASTn search like this one, where I search with a protein and I match the protein translation of a genomic sequence, I can click that graphics link and that will take me directly to a view of that genomic sequence in our graphical sequence viewer, like this one. And there is my protein match there. There's a number of mismatches there. You can see that at the bottom of my screen here. There's sort of these red marks or mismatches, but that's still a very good protein BLAST match. That's a tBLASTn alignment of the query against the subject sequence, which is this whole genome shotgun sequence from this particular bacterium. And in fact, if you check these, the annotation of these genes on either side, they are similar to the ones that I showed you a minute ago, although one of them is labeled a hypothetical protein. In fact, if you take that and do a BLAST search with it, you'll see that it matches one of the other two things. But the site-specific methyltransferase is annotated in the right way. So the genomic context in this particular case is conserved, and we saw it simply by clicking one link on the BLAST output. Now, what a lot of people, though, do when they do these kinds of searches, or protein searches, and in part, you really, to do a tBLASTn search against a large set of bacterial genomes is kind of burdensome, and it takes a long time, and it may fail for various reasons. So protein is more likely to work. The problem is the graphics link here doesn't do what you might think it does. It doesn't do what we saw a minute ago. It takes you to the protein sequence graphic, which doesn't tell you about the genomic context. However, there is another link that's often shown on the right-hand side when there's more than one protein available, in this thing called the identical proteins link. And notice that this is a particular kind of protein accession number that starts with a W and a P. This is one of our non-redundant RefSeq bacterial proteins. I'll talk more about that in a minute. So if I link to the protein itself, in our sort of Entree system protein web database. I can see a link to identical proteins there. And I can follow the link directly to the IPG report. And this is a list of, first of all the proteins that are identical. There may be in several GenPept submitted sequences that are identical to this reference sequence that starts with a WP. But the WP is annotated on a number of different genomic sequences. And so you can see those in the second column of the table on the right-hand side of this slide. And if I follow the link to one of those, it will take me to that coding region, the CDS region, in the nucleotide database with that particular range and that particular strand of the DNA focused in on. So that will give me a way of looking at the corresponding coding regions in all of these bacteria that have this protein annotated on them. So for example, this is the protein, a link from the protein that we're going to be looking at this genome in a minute anyway. This is a Salmonella enterica. Assembly, I'm just looking at this 3,000 or so base pair part that corresponds to the coding region of the blast match. And if you want to try to look at that view yourself, the link is there in this slide. And if I follow this to the genomic view, I can just manipulate the graphics view a little bit to the graphics view is what I clicked on here. And I can just take a look at the things surrounding that particular coding region. So that's the one that we were zoomed in on a minute ago, and that's labeled as a site-specific DNA methyltransferase, which is exactly what I would expect my blast search to match. Now, remember, we're not going directly from blast, so it doesn't show you the alignment in this particular case because we were just aligned protein to protein. But I'm looking at the region on the genome that corresponds to that protein. Upstream of that is that ATP-dependent helicase that we saw at the beginning when we were talking about sort of the canonical version of this. And then on the other side is that type 3 restriction protein, the binding site unit. So that was conserved. We saw that by following the links to the identical protein report and then to the graphical sequence viewer. So that's how you would do it on the web, and I'll demonstrate that stuff live in a few minutes. The other thing I want to mention is that you can access these identical protein group reports using the API. In particular, we're going to focus on using EDirect to do that. And we have a number of webinars on EDirect, so if you want more background on EDirect, you can go to our YouTube playlist, and the link is there on this particular slide. What EDirect is a Unix command line version of the Entree utilities, or the eUtilities, which is the API for our Entree system. It lets you search and retrieve records from all of our databases. It has processing scripts and an XML parser called extract. And each program outputs XML. And you can pipe it into another EDirect program or a Linux Unix utility for processing. Okay, and we've talked about this a number of times in various webinars, in fact, one last week. And certainly take a look at the documentation, which is very extensive and explains a lot about using EDirect. So I won't have time today to talk a lot about that, but I want to just point out some things that are useful that you can do with these particular reports. And we're going to be focusing on eFetch and then the program extract to look at some various things. So the format IPG argument to extract will give you that identical proteins group table that we saw a minute ago. And here's an example of how I could retrieve that and get the IPG report on the command line. The other thing that's going to be useful to us if we want to get the genomic context once we know what sequence we're going to be looking at is we want to use a particular kind of XML that's called GBC. It's a special XML format that has sort of these wrappers or built-in extract functions that let you parse out features from the feature table. And for example, this rather complicated looking command line here, at the bottom there I'm retrieving this record from the nucleotide database in the format GBC. I'm passing it to extract, and what extract is using this INSDC argument to look at this CDS feature. And within that feature, there are these various qualifiers that you can output simply by giving their names. So I can get the interval, and I can get the protein ID. So we can use that in a very crude way today to get that information. So here's what we're going to do. We're going to go to the web and do some web blast stuff. And really, I'm not going to run the searches. I'm just going to show you the results because I've already run them. It'll save us some time, but I'll show you how to get to the graphics view and how to do the things I talked about. And then we'll talk a little bit, sort of touch on using EDirect to get these things out using the command line. I'll show you how to do it with a single report. And then we'll sort of begin to talk about how you would do it with blast output. That's a more difficult problem because you have lots and lots of IBG reports in there. There's one for each protein, and each one of those could have many, many corresponding genomic regions. Ultimately, in some cases, if you wanted to do this in large quantity, you probably want to go to the assembly database and download the corresponding GFF files or the annotation files for the particular assembly. And I'll talk about that more in just a minute. So anyway, what we're going to do with this is we're going to get these nucleotide records and we're going to look at the neighboring genes, one up, one down, to see if they're the same or they're conserved, just like we saw a minute ago in those screenshots of the web. Okay, so let me escape out of the slides. And what we're going to be doing for our blast searches is I sort of went through a whole genome shotgun assembly of a metagenomic project, and I found an open reading frame in there that corresponds to one of these methyltransferases. And so we'll use that as a query sequence when we're doing our blast search today. If you want to see what that project is, the link is here in this handout. Okay, so the first things we're going to do is just take a look at the web searches. And remember, we did a search against the nucleotide database, and the one that I did was this tBLASTn search against representative genomes. So all I'm doing is just taking that RID, which I've saved. And this should be valid for several months because I preserved it. It won't be valid forever, but it should work for the short term. And this is a tBLASTn search, so this took a few minutes to run. But you can see there are all these hits to complete genomes. And we can pick any one of these we want to look at the corresponding graphical view to see, whether we think this is conserved. And actually, I'm just going to pick one that I haven't looked at before. So let's see if we get the same results here. So I'm going to take this chlorobium complete genome here. And let me just jump to that in the alignment section. And there's our tBLASTn alignment. But now if I click on the graphics link here, either one will give me the same thing because there's only one hit to this particular genome. And so it looks like we have some differences in annotation than what we saw a minute ago, which is fine. This is the kind of thing that you can identify by looking at this. So it did identify this open reading frame here as a site-specific DNA methyltransferase, but then there are also some things that are sort of part of that match that come up as annotated as separate proteins. Now that may or may not be correct. But so we already are seeing some genomic context of the blastocysts here. There are 2 genes annotated in this region. I can zoom out to see what else is around. So here's the gene that's immediately to the 5' of that. It says that's a transposase. Again, that's different than what we think of as the canonical version of this, but this is interesting, and it does give you the genomic context. And this is another different gene there, so we can keep looking around if we want to see what's available. Now, that's another case. Here we have, it looks like we have an extra gene here, but the next gene downstream is that type 3 restriction in nuclease subunit like we expected to find. And so this is an interesting kind of finding, and if you were interested in the biology of this organism, this would be useful for you to know. Okay, so that's that blast result. I'm going to close the graphical overview here for that one. And let's go ahead and get the other kind of blast results, with those protein results that we talked about a minute ago. And in this case, I blasted that protein sequence that we saw against the RefSeq protein database, which will give me entirely, for bacteria anyway, it will give me entirely WP style records. And so you can see that our methylase has this nice type 3 restriction methylase superfamily modification here, so that's good. That's what we would expect. And we have hits to lots of WP-style proteins from lots of organisms that you probably recognize the names of. Here's one, Salmonella enterica. And this is the one that we were looking at in the slides a moment ago. So let's go ahead and get that one. So I'll jump down to the alignment section here. And so I don't see the, I'm not sure that this is just a bug today, but the particular link to the identical proteins is not there, but it doesn't matter because I can obviously get it by going directly to the record itself. And that's a bug, but let's see if we can still get the identical proteins report. Okay, that's fine. Okay, so we do have the identical proteins report. This is a nice table that lets me see the coding region. And so I can pick a particular salmonella that I want, like right here. I'll just do the first one. And again, we're zoomed in looking at this particular coding region on this genome. I can go to the graphical view here. And there's my protein. And as we already know, because the protein says so, it's a site-specific DNA methyltransferase. But now I can zoom out a little bit and see what's there. And of course, we've already seen this in my slideshow a moment ago. So there's the ATP-dependent helicase. There's our methyltransferase. And there's the restriction binding site right there. Okay, so those are the two things that you can see in BLAST and how to get the genomic context for these matches. Okay, now let's go, and we're going to switch now back and forth between this sort of page here, and we're going to start using EDirect. And so this will be fairly quick, but you can always come back to this and get these command lines if you want to practice using EDirect. So let me just grab the first one here. First of all, I'm just going to get the IPG report so you can see that it's the same thing. So I'm going to search the protein database for this protein record, the one that we saw a minute ago, and I'm going to ask for the IPG format. And so we get this nice report here. It's just like the one we saw on the web. One of the things I want to do is to focus on the refseqs that are in here. Notice that there is an entry here on the table that says INSDC. That's a GenPep record, and it's identical to these refseq proteins that are in here, these WP accessions. It's all just one WP. So what I want to do is to clean that up and just get the refseqs. I can just use awk, which is sort of a Swiss Army knife for dealing with have the limited output. I can say, just give me the refseqs. That's going to be in column 2. And what I'll print out of that is simply, let me go ahead and clear this. The nucleotide accession numbers. So what we can do with those is we can go back in and we can retrieve those in that other format I talked about and go ahead and parse out the feature table to get the flanking genes on either side of this. And this way that I'm suggesting doing this is not a very sophisticated way, and certainly you may be able to think of better ways to do it, but it certainly makes the point that it can be done. So here's what I'm going to do. There's a lot of sort of extra business in here. And I'll sort of talk about what's important for this particular command line here. Okay. So we've already done this. I'm going to get the WP in the IPG format. I'm going to get out just those nucleotide accessions that are in column 3. I'm just going to pass the first two just to show you quickly what happens. But then I'm going to pass those two things to eFetch. That's what this XArgs business is. And that's very well discussed in the document that goes with EDirect and how to use it. So I'm going to go to NuCore and get these guys from the NuCore database in the GBC format. And then I'm going to use that extract command to get all of the coding regions. And then I'm going to do something really crude, basically just use grep to get that particular coding region, the one before it, and the one after it. So let me do that. So those are the first two, and they are indeed exactly the same. The ATP-dependent helicase, the site-specific DNA methyltransferase, and the type 3 restriction protein. And you could even extend this a little more to get, 2 up, 2 down if you want to. And that's what this next command line does. There's nothing particularly special about it, except I just changed the grep parameters. And instead of running that, I'll just show you what the result is. Okay, and so you can see the only real difference, so I've got 2 up, 2 down here. The only real difference you'll notice if you look through, sometimes they're flipped around because the particular coding region is on the opposite strand. And so the order is sometimes different, but the genes are the same. Unlike we saw a minute ago in the blast where they were not the same, but in this particular case they are. These are all the salmonella assemblies that we saw a minute ago. Now, what about doing that for a blast result? So what I did was I took one of those blast searches that we did a minute ago, and I saved it from the web onto my hard drive, and then I can go ahead and search that and get the IPG reports, and that's what this does here. It basically parses the blast report, and we have a webinar that's all about doing that, if you want to see that. So basically what I'm getting is all of those. I just did the first 10. That was 100 different blast things. And so you just got the basic IPG reports for all of those. It's kind of confusing because it wraps the the lines are too long, but you can see that these are the IPG reports for all of them. Now you could parse this with a script if you want to and do the same kinds of things that we did before with the other ones. The only issue is you've got to now have a different WP, and you'd have to go through and write a script to handle this in a more sophisticated way. So, I can rewrite this if I want to. I can get this in a little bit more human readable format where it gets the assembly accession sort of highlights that for you. And that's sort of the next sort of way to do this. And I'm not going to go through all this, but you're certainly welcome to take a look at this and see the sort of strategy. The idea here is to go ahead and get the assembly out of the last column that would be printed out here. And I'll go ahead and show you that file. Then there are some strategies that are documented fairly well on the Genome's download about how to download particular files from the assembly directory. The file that I would recommend you do this with is the GFF3 file for each one of these. And then it's a very simple matter because it's a single line per coding region, and you can parse those out of there pretty easily. So I'll just show you that RefSeq IPG report here, and then we'll stop for some questions. So that's just, I used awk to sort of simplify that report a little bit. But you can continue along those lines and go ahead and go do a search against the assembly database using EDirect to get the path to the FTP directory that contains the assembly files. You could write another script or make a more complicated script that could go ahead and pull down the GFF file for each one of these and parse it for the particular proteins of interest. But of course, you have to have each a different protein for a particular one of these assemblies sometimes. Okay, so I'll stop there and see if anybody has any questions, and we'll leave it open for a couple minutes in case somebody thinks of something at the last minute. So what we saw is that you can link to the IPG report to get the nucleotide coding region for WebBlast. You can fetch and parse protein IPG reports using that GBC XML with EDirect and other command line tools for a single report or for the standalone blast output. And the last slide has a number of URLs. Many of these I've mentioned in the slides earlier, but you might want to download that and take a look at that if you want to visit any of these things. In particular, take a look at the YouTube playlist for EDirect and the EDirect manual if you're interested in doing more things with EDirect.
Hi, Peter. We have one quick question at this point. One of our users is interested in how we can get the T-blast end results in the form of protein sequences using the web, not EDirect.
So you want to get the translations of the genome. Right, he wants to get the protein translations. So where there are matches like the ones I saw where you are matching annotated genomic sequences, yes, you can do that because you could do the same kind of sort of process where you could go and find the annotated proteins. However, if you're talking about doing a T-BLAST-N search against something like a WGS record or another kind of unannotated nucleotide sequence, there is no way for you to get the corresponding coding regions. You can take the nucleotide sequence, which is what I did to make that protein that we used as a query, and go to our OR finder and do a 6-frame translation on that to get the particular open reading frame that's on it, if that's what you wanted to do. So I guess that's the answer. You'd have to take the genomic DNA hit and do some kind of translation yourself. The OR finder on our website will let you do that. And I can quickly show you where that is in case you don't know. So it's just the NCBI URL and then OR finder as a single word. And you can put your open reading frame or an accession number right in here to generate, and it will generate the protein sequence for you. It doesn't do it in bulk, and I don't think it does it in a way that will let you download these things, but we can check that out, and I'll put the more elaborate answer in the Q&A document that goes along with this. Ron, is there anything else?
The same person was interested in how to translate 2,000 hits using the web, and that's not possible, but we can put an example in the documentation after this webinar on the web, on the FTP site, as to how you can actually get your protein sequences out. There's another question that somebody had, which is he's interested in IPGs, but he wants to know if you can filter the IPG clusters shown to those that have specific sets of organisms. And he's interested in EDirect or, using scripting. Can you filter the IPG hits that are returned to those that only have a certain group of organisms?
So I think if I understand the question is yes, you can. You can actually search IPG, and I'm not positive you can do. You can search IPG on the web. I haven't tried it really on the command line, but it ought to work the same way. You can put an organism query in there, and it will give you the clusters back, the identical protein groups back that have that particular organism in them. I think that answers the question, but I'm not sure. There is a separate, IPG actually has its own interface that you can search here. We weren't really talking about this database per se today. We were talking the link to this database and the format that you can get for from the protein side. But there is a way to search the IPG reports this particular way. Okay, well, I think I will close then with that. Thank you very much for coming, and I'm going to end the webinar now.